Good morning, everyone. So today I'm happy to talk about our study. It is actually a long-term study focused on revegetation and ecorestoration of mine tailings facilitated by Arbuscular mycorrhizal fungi and organic waste byproducts. So when it comes to revegetation of tailings, it sounds like a cost-effective and environmental friendly solution as a remediation option for these tailings, but actually it is very challenging. Otherwise, a plant can colonize these sites, but in fact these sites remain barren for decades. They cannot support plant growth. So in our approach, we kind of focus on correcting multiple factors involving like the selection of candidate plant species and also the appropriate amendments. And also amendment strategies to prolong the benefits. Unfortunately, some of the amendments, they are beneficial, but only for short term because their benefits can dramatically decline with time. And field scale failure scenarios have happened due to this issue. So usually short-term studies, the results can be deceivable. And also amendment strategies like involving the use of powerful microorganisms such as Arbuscular mycorrhizal fungi. So for this study, we used tailings collected from Viburnum mine 28 impoundment. And actually, you can see this is the tailings. It's very poor structure. And also the characteristics, very poor general soil characteristics, very low cation exchange capacity, organic matter. That means there is no microbial activity, lack of essential nutrient. And also on the top of the phytotoxicity, we have a high concentration of lead, zinc, and copper. So this is actually a long-term experiment because we wanted to test the long-term feasibility and sustainability of our approach. So what we used, as you see here, we used 6 different treatments, and all of this actually tailings. We only amended the top layer. We amended with either biosolids or a combination of biosolids and biochar, or we added the mycorrhizal fungi. So, biosolids rich in organic matter can provide essential nutrients, but the problem with them, they decompose very quickly, and all the benefits can go away just with time. But biochar resists decomposition and has specific retention capacities, especially for metals. But also, we incorporated the mycorrhizal fungi. We wanted to promote ecosystem restoration and soil building over time. And the goal was, as I said, to support long-term growth, and especially native growth, like especially here native to Missouri. And also, are we building soil over time? So are we also transferring the benefits from the top amended layer to the unamended lower tailings layers? And actually, here is the results after 7 years growth. So what you can see here, as you can see, it is very obvious how only incorporating like mycorrhizal fungi with the biosolids in that top layer impacted the root to growth. So on the picture on the left hand, actually the root barely penetrated the lower layer. Compared to on the right hand, we have extensive root growth, especially in the lower layer. Then what we collected some soil cores from each layer to be tested for microbial community and also for organic carbon as indicators of soil formation. And this actually, just to show you the growth, that was the first year growth. And this is actual annual species. We tested 5 different species in this experiment. And you can see for the first growing season, you can see you have the two treatments. On the left hand, there is no mycorrhiza, but the rest of it incorporating the mycorrhiza, how significantly impacted and improved that plant growth. And actually, this is the second-year growth. And this is annual species, as I said, and actually this is the second-year growth was from self-reseeding. But we stopped for this species at this time. But it is obvious the impact of incorporating mycorrhiza fungi on the growth of this species. And this is another species. This is, you can see, the first growing season. You can see the same impact when we incorporated mycorrhiza fungi compared to non-mycorrhizal, the two treatment on the left hand. And here is the 7 growing season after 7-year growth. So incorporating mycorrhiza, again, or even on this species, significantly promoted plant growth. Here is another species, purple prairie clover, and I want to note all of these species, they are selected based on their ecological value. So we wanted to promote, in the meantime, ecosystem restoration. We don't want to, okay, we establish vegetation, but we end up with just monoculture or invasive, dealing with invasive species. So we're trying to use natives and promote ecorestoration on these sites. And this is after 7-year growth. On this one, actually, this is nitrogen-fixing species, so they can kind of use atmospheric nitrogen for their own growth, but can also store within their root nodules and can fix nitrogen to build soil and support other species. So you can see in this one, actually, even with non-mycorrhizal species, kind of still supported good growth after this long-term growth. And here is another good species, bundle flower. I don't have for the first year a picture of this because we used to image this individually, but this is really good species and supported good growth after this long-term experiment. It is the impact of mycorrhiza fungi, actually, it is more apparent here in this grass. This grass, non-fixing nitrogen species. So you can see this is the first season and compared to the 7-year growth. So you can see without mycorrhiza fungi did not actually support the growth of this species. However, the impact of AMF more pronounced here. Then we tested, as I said, we tested for mycorrhiza, for microbial community. As an indicator, we specifically used BLFA analysis, which is a molecular component of microbial cell membrane. And we used this as an indicator of soil formation because it is particularly good when we want to study the health of the soil. It is linked to the soil health. So, as you can see here, actually we here only did it for 4 treatments because we're limited with resources, so only 1 mycorrhiza culture we tested here. In our experiment, we had 2 mycorrhiza culture isolated from different areas. So what we can tell from this one, total BLFA, total microbial biomass, and here, based on this analysis, it is more pronounced in the lower layers, unamended layers. So actually, especially in the green bars, which if we map them here, you can map them to these soil cores. And what we notice here, we notice like this promotion of microbial growth is driven by roots, right? Because this is the roots, as you can see, the roots with mycorrhiza fungi, there is extensive root growth, especially in that layer. So we think like roots acted as vector transferring microbial to the unamended lower layer, which is really a good sign of soil formation. And here is the organic carbon tested in these soil layers. We wanted to see after this long-term experiment, are we adding any carbon to these layers? And it's kind of the same trend as total BLFA. As you can see here, also, it looks like mycorrhizal fungi promoted carbon sequestration within that deeper unamended layers, the green bars, and if we map them, it is also, we can say, linked to the root growth in that layer. So, subgroups of microbial community within that analysis, and specifically those groups, they are linked to soil health, especially for organic matter decomposition, for a nutrient cycling, so they are very good indicators of soil health. And we can observe the same impact of mycorrhizal fungi, especially at the unamended tailing layers, lower layers, which is really a good sign. Now this is the mycorrhiza fungi itself. So we added the mycorrhiza, we inoculated only the top layer. But we can see like mycorrhiza, I mean, it is not surprising, like we find it in the blue bars that would represent the top layer because we added there. But we can still see green bars, again, like mycorrhiza fungi transfer to the unamended layer. And actually, the structures, what you see of mycorrhiza fungi, so what they do, they colonize the plant roots, and from there, they extend their structures, particularly hyphae, so they can increase, they actually modify even the root architectural, but they can increase the root absorption area, they promote growth through like more water uptake, more nutrient uptake, but also they can help with soil building over time. So now this experiment was based on bat experiment, right? And it's we have very controlled size. And what happened, like plant roots hit the bat wall and then accumulated within that lower layer. But we expect the benefit to be different in their field setting. Because, you know, a plant root can freely grow horizontally and vertically, which means like we can explore larger volume of soil or tailings. And that means the benefits associated with the mycorrhizal fungi can be expanded to a larger volume. And actually, we did start a test plots study. We translated our findings from the greenhouse to Sweetwater Mine. It was in spring 2023. And this is some of the, what you can see here, some of the results there. We have ongoing work. We are assessing the impact of mycorrhizal fungi and waste byproducts on soil health and their field setting using our test plot established at Sweetwater. And also, along with that, we are working on experiment assessing the impact of reprocessing of mine tailings, like when we use it as a source to extract critical minerals. Okay, the generated waste, the environmental impact of it is still unknown. So we're trying to address this question by characterizing and also assessing the ecological and impact and environmental impact of reprocessing through like phytotoxicity studies, leachability, and also ecotoxicological tests. And with that, I would like to acknowledge my mentors and collaborators, and particularly my advisor, Dr. Joel Birkin, and also soil lab testing at Mizzou, and also specific thanks to Chris and Jen from Doran Company. Without their support and assistance, this work wouldn't be done, and also funding from the Doran Company and NSF. And thank you so much, and I'm open for any questions.